Good morning, everyone. I hope you're all doing well today. This is a first for me today, presenting at an online conference, so I'm hoping all this works. I wanted to begin by thanking the Macular Society for inviting me to speak to you all today, and to all those involved in the Macular Society for supporting our research into vision loss. Now, the Macular Society has asked me to give a talk about the research that I do, which is working on a group of diseases called bestrophinopathies, and I do this using stem cells. So I'd like to give you an overview of this research and take you through what a stem cell is and why they're important for our work. I'll talk about how patients and stem cells are helping us to understand the causes of blindness, before going on to discuss how we're using stem cells to test new therapies for inherited eye disease. Now, I've been working on macular disease for nearly 15 years, and the majority of this time has been spent using stem cells. So what are stem cells? Well, they're specialised cells that can develop into the many cell types in the body, from muscle to blood to eye cells. And there are a couple of different types of stem cells that you may have heard of. And at the top of the list are so-called pluripotent stem cells. And these are cells that can make any cell type in the body. And we find these in embryos at a particular stage in development, and this small group of cells in the embryo is capable of growing into every cell type needed to create a whole body. Then there's also what we call adult stem cells. Now these adult stem cells can make some, but not all cell types, so we call these multipotent stem cells because they can make multiple cells. So for example, the stem cells that are contained in your bone marrow make all the different blood cell types in your body, but they're unable to make an eye cell. And so multipotent adult stem cells can be used to sort of naturally replace the dying cells of your body and regenerate tissues, say like skin after injury. And then we have what we call fully differentiated or specialised cells, and these have a specific role in the body and they don't divide. And these cells have special properties that allow them to do a specific job in the body. So for example, the photoreceptors in our eye allow us to detect light and enable us to see. So if we have all of these stem cells in our body, why are we not able to repair certain tissues when they're diseased? Well, macular diseases affect a small region at the back of the eye called the macula. And there are many cell types in this region that are needed for vision, including photoreceptors that detect light, the retinal pigment epithelial cells, or RPE, that look after the photoreceptor cells, and then the retinal neurons that help transmit the visual information to the brain. And this region is exposed to many daily stresses. So this area is very vulnerable over our lifetimes, and vital cells like the retinal pigment epithelium can become worn out with age or because of specific genetic problems. Now, unlike other regions of our body, such as the skin, those specialised cells at the back of the eye aren't able to regenerate, and there are no stem cells in the eye that can make new cells to replace them. So the RPE keep the photoreceptor cells healthy. So if the RPE die or degenerate, so do the photoreceptors, and this results in irreversible vision loss. Now, I've been working on the retinal pigment epithelium with Professor Pete Coffey and Linda de Cruz as part of the London Project to Cure Blindness. And for this project, our aim was to make new retinal pigment epithelial cells, or RPE cells, for patients with age-related macular degeneration. Now, as one of the researchers in the lab, I was involved in working out ways to make new RPE, and we decided to use human embryonic stem cells and worked out a way to turn these into RPE. And we grew the replacement RPE cells on a membrane as a patch of cells that could then be transplanted into the back of the eye. And this project developed into a clinical trial which has successfully treated 2 patients with age-related macular degeneration. Now, during this time in the lab, a new technology emerged called cellular reprogramming. And to say this was an amazing advance in stem cell science is a bit of an understatement. This Nobel Prize winning work has completely changed the way that we can study human disease now. And now cellular reprogramming is a way to take a specialised cell, like a blood cell or a skin cell, and turn back time, rewinding their clock and turning them back into a pluripotent stem cell. Now, we use a special method to induce pluripotency in these cells, leading to their name, induced pluripotent stem cells, or iPSCs. So these cells are like human embryonic stem cells. They can be turned into any cell in the body, and we were quickly able to show that we can make RPE from them in the same way that we did for human embryonic stem cells. Now, being able to reprogram cells led to a whole new area of research involving patients with inherited diseases. So we can take a skin biopsy from patients who volunteer for our research. The biopsy is then cut up into very small pieces and put into a tissue culture dish. And over a couple of weeks, cells called fibroblasts grow out from the biopsy pieces. And we use these fibroblast cells for cellular reprogramming. And in about a month, we get small groups of stem cells growing in the dish. We then take out these stem cell colonies and grow them further, and as long as we keep them in the right conditions, they're able to divide and make more stem cells. But if we grow them in certain conditions, we can get those stem cells to turn into retinal pigment epithelial cells. Now, these patient-derived cells are important for eye research for two specific reasons. The RPE cells we create from patients contain the same genetic information as the patient. So when we grow eye cells in a dish, they can contain the same mutation that causes vision loss. So we can use the cells to look at what's happened during the early stages of disease in a dish. And by comparing RPE cells created from people with no disease, we can start to understand what has happened in the cells to cause vision loss. And secondly, because these cells come from patients and contain the same genetic mutation as the patient, we can use these RPE to develop and test new therapies for treating inherited sight loss. Now, there are various inherited diseases which cause blindness, and these can all affect the different types of cells in the retina. So we've started a stem cell bank at the UCL Institute of Ophthalmology to store induced pluripotent stem cells that we've created from patients at Moorfields Eye Hospital. And we now have over 40 different patient cell lines which are being used by various groups at the Institute to work out why patients are losing sight. So having these cells from patients and using them to create eye cells has transformed the way we could approach studying macular disease. So I'm going to give you an example of some of the work that we've been doing working on bestrophinopathies. Now, bestrophinopathies are a group of inherited diseases that affect the RPE cells at the back of the eye. Now, this group of diseases includes Best disease, autosomal recessive bestrophinopathy, adult onset vitelliform macular degeneration, and autosomal dominant vitreoretinochoroidopathy, or ADVRC. Now, inherited diseases are caused by changes in our genetic code, and these are known as mutations. Now, bestrophinopathies are classed together as a group of diseases because they're caused by mutations in a single gene called bestrophin-1, or we call it BEST1. Now, the BEST1 gene is a region of our DNA that contains the instructions to make the BEST1 protein. Now, the cell machinery responsible for making the protein reads the genetic instructions for BEST1 and then produces the protein. And the BEST1 protein is only made in the RPE cells of the eye, where it forms a channel along the base and up the sides of the RPE cell membrane. And its role in the RPE is to regulate the movement of chloride molecules in and out of the cell. Now, changes in the BEST1 DNA sequence can change the instructions to make the protein, and this leads to changes in the way the protein works, which can have a big impact on cells. Now, we know that there are over 250 disease-causing mutations in the BEST1 gene, but these mutations seem to have different effects and can cause different diseases. So we've been looking into how different mutations can cause bestrophinopathies, and we found a number of things in different patients. As I said earlier, the BEST1 channels are normally found along the bottom and up the sides of the RPE cells. Now, in some patient cells, the BEST1 proteins form a channel which is found in the right place in the cell, but it doesn't work properly. And in some patients, the channel is found in the wrong place. For example, the BEST1 channel is normally found along the bottom and up the side of cells, but in some patients, it's also found along the top of cells. And in certain patients, the mutation can cause no bestrophin protein to be made at all. And in other patients, the protein doesn't make a channel and instead hangs around in the inside of cells and generally causes a bit of a nuisance. So these changes in function and location, or even in the complete loss of bestrophin-1, can affect the fine balance of processes that occur in the RPE cell. And over a period of time, this can cause the cells to become unhealthy or even to die. And ultimately, this stops the RPE cells from keeping the photoreceptor cells healthy, which can lead to their death and therefore loss of sight. So the induced pluripotent stem cells for patients are helping us to find out what is going wrong in their own eye cells, which means that we can identify early disease pathways that could potentially be targeted with therapies. But one of the biggest problems when we're trying to develop a therapy for inherited disease is that the issue still lies within the patient's own DNA. The cells will continue to make proteins that don't work properly unless the DNA is changed. And for a long time, a therapy that fixes issues with DNA has been a bit of a pipe dream. But there's been some incredible developments in both gene therapy and genome editing over the last few years. And this is now turning the pipe dream into a very real reality. Now, the eye has been at the forefront of these advances, and from a scientific point of view, the eye is the perfect organ in which to develop and test these new therapies. It's easily accessible, and the front part of the eye is clear. It lets light in, so we can easily see through the eye to the back of the eye into the retina that's affected by disease. The eye is pretty isolated from the rest of the body, so we can inject therapies directly into the eye to the region that's affected. And because we can administer these therapies directly to the macular region, we only need to administer very small amounts of the therapy. The eye also has an immune privilege, and this means it's able to tolerate the introduction of new cells or medicines without developing an immune reaction that you would normally see in the rest of the body. And when it comes to finding out whether a therapy is safe and it works, the eye also gives us a unique opportunity of having the perfect control, because at the start of clinical trials, only one eye is tested in patients. So researchers can compare any changes in the treated eye to that of the untreated eye in the same patient. And the eye is also unique because it's an organ that we can see through, and we can use medical imaging techniques to look at individual cell layers at the back of the eye to find out whether these therapies are working to maintain sight. And ophthalmologists can also use very simple eye tests to see whether there's been any improvement or further deterioration in vision following treatments. So it comes as no surprise that the first NICE approved gene therapy treatment in the body was for an inherited eye disease. So what is gene therapy and what does it entail? Well, gene therapy is a treatment that provides a new working copy of a gene. The cells can then use these new genetic instructions to make a working copy of the protein. So gene therapy acts as a kind of supplement, adding additional copies if protein levels are low or absent. But what gene therapy doesn't do is it doesn't change the patient's copy of the gene. So new healthy copies of the gene are delivered direct to the RPE cells or the retinal cells by injection of a virus into the back of the eye. Now, I think over the last year we've all seen the impact that viruses can have, but viruses also have very special properties that enable us to use them medically to deliver genes. Now, viruses such as the common cold infect cells, inject their DNA and basically hijack the cell's own machinery so that the cell then produces viral genes and viral proteins, ultimately making new copies of the virus that can then go on to infect other cells. And for a long time, scientists have been using viruses to deliver genes into cells. We can modify viruses, so we can take out the genes that cause viruses to replicate and cause disease, and we can add in new genetic instructions for genes that we want a cell to make. Gene therapy is now available in the clinic for patients with mutations in a gene called RPE65, and this can be found in some patients with retinitis pigmentosa and Leber's congenital amaurosis. Now, the therapy, termed Luxturna, delivers a new, healthy copy of RPE65 into cells, and this can improve vision and help a patient navigate daily tasks. Gene therapies are currently being developed for macular diseases, and a recent trial has started in the UK for patients with age-related macular degeneration. The therapy has been developed for a gene called complement factor I and is being used in patients who don't produce enough protein. So it's hoped that by adding new copies of the gene, this will increase protein levels and reduce vision loss. Gene therapies are also in development for inherited macular diseases. For example, there's a group in the US who've recently shown that the symptoms of a canine form of bestrophinopathy can be reduced with BEST1 gene therapy.
So the future looks great. Gene therapy to treat macular disease is in progress, and we're hoping for promising results from the clinical trials and also from the basic research.
But what happens in patients that are producing 40 copies of the protein that doesn't work correctly or doesn't work in the right place, which can happen in some patients with dominant diseases? Gene therapy might not work so well in these patients. Remember I said that gene therapy doesn't change a patient's DNA. So if the cell is making a faulty copy of the protein, it will continue to make that faulty copy following gene therapy. Now, gene therapy will add additional healthy copies of the gene into cells, but if the patient's own faulty copy is interfering with the healthy copy, it could still stop the therapeutic proteins from doing their job. So we need to look for alternative treatments. And in the last decade, a method known as genome editing has rapidly transformed drug discovery for genetic diseases. CRISPR editing has become the most popular method to change DNA. It's a relatively simple system that works well and is easily adaptable. So the CRISPR system itself was developed from a naturally occurring viral protection response that's present in microorganisms like bacteria. So when bacteria cells are infected by a virus, the bacteria creates a record of the viral DNA in a way that's similar to the way we make antibodies in response to viral infections. So the bacteria stores this record of the virus in a region of its DNA called the CRISPR region. And the bacteria then uses this DNA record as a guide, so that the next time the cell is infected by the same virus, it can target the virus, become resistant to it. So the guide takes an enzyme called Cas9 to the viral DNA, and once the guide and the Cas9 enzyme are bound to the viral DNA, the Cas9 enzyme can then cut this foreign DNA, and this effectively disables the virus. So rather than using CRISPR system to cut viral DNA, scientists have adapted it to cut any DNA sequence, including human DNA. And to do this, what we do is we create a synthetic guide, which is designed to bind to a specific region of the human DNA sequence. So if we're trying to target a mutated region of a gene, we would design the guide to be somewhere near that mutated region. We can then deliver the guide and Cas9 enzyme into the affected cell, where they'll join together and they form a complex that goes around the DNA and searches until it finds a region that matches the guide. When it finds this corresponding region, the CRISPR-Cas9 system acts as a pair of molecular scissors and cuts the DNA at that point. And scientists can direct the CRISPR system to break DNA at any region by just modifying the guide sequence to bind to that region. And then we'll cut there. So breaking the DNA is the first step in genome editing. And once the DNA is broken, it then needs to be fixed. And the cell has a number of ways of doing this. And at the same time, we can change the DNA sequence that's being fixed. So one way we can do this is by replacing the mutated region. So we can think of this as kind of gene plumbing. So imagine a pipe that has a hole in it. This is equivalent to the mutation site. We have to cut the pipe around where the hole is. We then have to put in a piece of replacement pipe and weld it back together.
We can do a similar thing in gene editing. So once we've used CRISPR to break the DNA, we can then give the cell a new replacement copy of the DNA, but this time the replacement copy doesn't have the mutation. So when the cell repairs the break, it incorporates this new piece of DNA into the site, replacing the mutated region. And replacing this affected region of the gene is a permanent DNA fix in cells. Patient cells will use the fixed copies of the gene to make healthy copies of the protein.
And we can also use CRISPR to switch off a gene. And to do this, we create the DNA and then let the cell's repair mechanism join those broken ends back together. And this is a quick fix repair in cells, and the mechanism isn't perfect. And it can lead to additions and deletions in the DNA sequence, which essentially creates a nonsense set of gene instructions. So this makes either incomplete or non-functional proteins. So essentially we're switching off that gene in those cells. However, gene editing to repair or switch off a gene might not be the best approach for dominant diseases where there's a faulty copy and a healthy copy of the gene present in cells. So with the help of the Macular Society, we're working on a different method to edit the DNA, and what we're attempting to do is to switch off the faulty mutated copy of the gene and leave the healthy copy able to do its normal job. And to do this, we're looking for small differences in the DNA sequence of patients that have mutations in their gene. So these are called variations. They're changes in the DNA sequence, but they don't cause disease. And the copy of the gene that we inherit from one parent can have different variants from the gene that we inherit from the other parent. So we sequence DNA in patients to identify these variants, and importantly, which variants are present only on the faulty copy of the gene. So we can then target CRISPR to cut at these sites only. And this allows us to target and cut only the faulty copy of the gene. And our approach uses two guides to cut the DNA, and this will enable CRISPR-Cas9 to cut at 2 points in the gene and excise a huge chunk of the gene. And when the DNA is repaired, this chunk of the gene is removed, which will inactivate the faulty copy of the gene, leaving the healthy copy to produce healthy bestrophin protein that will do its normal job. So I suppose I'd better get back to the main theme of the talk, which is stem cells. And these cells are really important for the development of the new therapies. So we have patients with their mutations that cause disease. We've taken their cells, we've turned their cells into stem cells, and then turned these stem cells into the RPE cells. So we now have the perfect system to test our new therapies, and that's what we're doing. We have patients where we know their mutation. We have sequenced the whole of their bestrophin gene and have identified variants on the normal copy of the gene and variants on the faulty copy. And we've designed guides that will only cut the DNA on the faulty copy. So we're currently testing these guides in our patients, and just after Christmas we introduced our guides and the Cas9 enzyme into our patient RPE cells and found that indeed they did cut out the faulty region of the BEST1 gene. And then, unfortunately, we went into lockdown and had to stop our experiments. And now we're slowly starting to get back to work and opening up the research labs. So our next step is to see whether the genome editing which we performed has worked. So we want to see, is the healthy copy of BEST1 going to the right place in the cells? Is it doing its normal job? Is it transporting the chloride ions into the cells? And what effect does gene editing have on the function of cells? Are they now behaving normally? So there are all these questions we can answer using stem cells. So essentially what we're doing is a clinical trial in a dish to see whether the therapies work in cells. So this research would not have been possible without support from the Macular Society, and we would like to thank the Society and all its members for your support. And over the last few months it's been difficult not to be able to get into the lab, so we've tried to focus on other projects that we hope will help patients. And one of my students, Sim, has created a website resource for bestrophinopathies, which looks into disease, how it's identified, and research into treatments. And although it's designed for patients with bestrophinopathies, it covers many areas which may be useful to patients with other inherited diseases. So I'd encourage you to visit our UCL website to find out more. And you can either access it using the QR code here, so you can just point your phone camera at the code and it will take you directly to the website, or you can access it through the address which is bit.ly/best-resource. And with that, I'd like to thank you all for listening, and I'm very happy to answer any questions you have. Thank you.